My name is Nicolas PAVILLON, and I work as a lecturer at the Biophotonics laboratory at IFReC. The purpose of our laboratory is essentially to develop new techniques to observe cellular processes, in particular in the field of immunology, especially by employing optical technology. This means that we are really at the interface between physics and engineering, where we take the tools to design our systems, and biology and immunology, for which, of course, then we apply these techniques to study relevant biological processes. This means that one of our the main part of our work is to actually build the systems that we will then use. In my case in particularly, I'm employing techniques called, for example, quantitative phase microscopy and Raman spectroscopy, which are able to provide us both morphological information of the cells based on the imaging, but also chemical information based on spectroscopy, where we can retrieve information about the constituents of the cells, such as sugars, proteins, DNA, and derive from this information what is actually happening in the cell. Especially we have used this research to extract hundreds of parameters based on these two techniques, and such measurements which we can then apply on thousands of cells in order to retrieve relevant information for biology are ideal candidates to be used in machine learning technology, which can then sort out this complexity in order to retrieve understandable scores that we can interpret, such as detecting the functional cell type or its activation state. And we have actually used this to detect the activation state of macrophages cells, where it's known that activation is a very complex process that can be triggered by multiple signaling pathways. We could find that such an approach that we are using can in fact work even better than standard cases that use staining to observe very specific molecules that are known to occur during inflammatory response. Whereas the techniques we use can be applied to live cells compared to staining, which has usually to kill the cells to make them observable. So we can observe live cells in this way, and as this technique is based on the observation of multiple molecules, we believe that such an approach can become even more accurate thanks to this overall observation. And as this technique is also general, we can apply it to different fields, and we are currently studying how we can apply it to study different immunological processes and cell behaviors for different cell types.